Imagine you're walking through your living room in the pitch black, right? And you violently stub your toe on the coffee table.
Oh, the absolute worst.
It really is. And you feel that physical impact instantly. But the actual, agonizing, throbbing pain, that takes a full, excruciating second to hit you.
Yeah, there's always that dreadful pause before it really starts hurting.
Exactly. Why the delay? I mean, why does your body process the physical touch in a fraction of a millisecond while the pain signal seems to just, I don't know, get stuck in traffic?
Well, it's a lag time that actually exposes the underlying engineering of our entire nervous system. We tend to think of our internal wiring as this uniform electrical grid.
Yeah, like a signal is just a signal.
Right, but that 1 second delay proves that our nerves operate on entirely different biological speed limits, and it's all based on a highly specific, microscopic infrastructure.
Okay, let's unpack this. Welcome to today's deep dive. You're in for a really fascinating one, because today we are breaking down the anatomical physics of myelin and nerve speed.
It's a huge topic.
It is. And our mission for you today is to completely dismantle the textbook definition of myelin. If you think of it as, just passive rubber insulation wrapped around a biological copper wire.
Which is how it's usually taught, unfortunately.
Right. But if you think of it that way, you are missing one of the most dynamic, metabolically active systems in the human body.
Absolutely.
We're going to explore how your nervous system engineered the speed of your thoughts and how it is actively remodeling itself right now, literally as you listen to this.
And to really understand the complexity of that system, we have to look at the physical problem of transmitting electricity through a biological organism.
Okay, set the stage for us.
So if you have an electrical impulse traveling down a bare, unmyelinated nerve fiber, it behaves kind of like a leaky, uninsulated cable in a wet environment.
Because the body is mostly water.
Exactly. The current leaks out across the cell membrane into the extracellular fluid, so the signal loses amplitude rapidly.
Meaning it gets weaker as it travels.
Right, which means the nerve has to constantly expend energy to regenerate the signal at every single micron along the way. That takes time. It's incredibly slow.
Biologically speaking, if an organism needs a signal to travel faster, say, to trigger an escape reflex before it gets eaten, nature really only found two physical workarounds, right? To decrease that electrical resistance.
Yeah, pretty much two main strategies.
Strategy A is basically brute force. You just widen the pipe. Increasing the diameter of the axon lowers the internal axial resistance.
Right, which brings us to the classic model of invertebrate neurobiology, the giant squid axon.
The famous squid.
The squid evolved to use that exact brute force method to drive its jet propulsion escape mechanism. Its axon is up to 1 millimeter thick.
Which is massive for a single cell.
It's huge. You can literally dissect it out and see it with the naked eye. And because of that massive diameter, the internal resistance drops enough that it can conduct action potentials at about 20 to 25 meters per second.
Which is fast enough to escape a predator, sure. But it comes with a severe structural penalty. I mean, is it fair to say that if humans used unmyelinated squid-style axons, it'd be like comparing an old, massive desktop computer from the 90s to a sleek, modern smartphone?
That's a great analogy. Because yes, if we relied on that brute force strategy, our spinal cords would have to be several feet wide just to handle basic motor functions.
Just to walk around.
Yeah. But the analogy actually understates the metabolic reality. It isn't just about the physical space in the skull or the spinal cord. It's about the energy required to maintain cells of that sheer volume.
So mammals went a different route.
Mammalian evolution opted for strategy B, which is elegant engineering over brute force. Instead of widening the pipe to lower internal resistance, we increase the membrane resistance. We wrap the wire in myelin.
And the dimensional differences are staggering here. A mammalian myelinated axon is only about 10 to 20 micrometers across.
Barely visible.
Right, that's up to 100 times thinner than the giant squid axon. Yet it conducts signals at up to 120 meters per second. So it's a fraction of the size, but 5 times faster.
And that evolutionary leap, that sheer space efficiency, is exactly why we can pack nearly 100 billion neurons into a human skull.
But achieving that kind of speed doesn't happen just by, greasing the biological pipe.
No, not at all. The addition of myelin fundamentally alters the biophysics of how the electrical impulse actually propagates down the axon.
Well, let's break down that mechanism, because the history of this discovery is super fascinating. We've known about this substance for over a century. Rudolf Virchow coined the term myelin way back in 1854.
Yeah, pulling from the Greek word for marrow simply because of how it looked under a microscope.
But he didn't grasp the electrophysiology at all. It wasn't until 1878 that Louis-Antoine Ranvier noticed a glaring structural anomaly. The insulation wasn't continuous.
Right, he observed these periodic bare gaps along the nerve fiber, which we now call the nodes of Ranvier.
And for decades, neurologists assumed this was a design flaw, didn't they?
They did. I mean, if myelin is supposed to be an insulator preventing current leak, why on earth would you leave microscopic stretches of the axon completely exposed to the fluid?
It seems entirely counterintuitive, at least until you look at the 1949 work by Andrew Huxley and Robert Stinfley. They proved this concept called saltatory conduction, from the Latin word saltare, meaning to leap.
But we should really clarify the physics here because the term leaping causes a lot of confusion.
Yeah, let me jump in on that because the electrical signal isn't literally flying through the extracellular space between these nodes like a spark across a spark plug, right?
No, not at all. The leap is just a functional illusion. To understand what's actually happening, you have to look at the distribution of ion channels.
Okay, break that down for us.
So, to generate an action potential, the nerve membrane relies on voltage-gated sodium channels. In an unmyelinated axon, those channels are distributed uniformly along the entire length of the nerve.
Like evenly spaced out.
Exactly. But in a myelinated axon, they are almost entirely absent underneath the myelin sheath. Instead, they are clustered at incredibly high densities exclusively at those bare gaps, the nodes of Ranvier.
So when an electrical impulse hits the insulated stretch of the axon, what is physically driving it forward without those sodium channels there to help?
It's a process called passive electrotonic spread.
Electrotonic spread. Okay.
Because the myelin sheath acts as a highly effective insulator with very low capacitance, it prevents the current from leaking out. So the voltage simply washes down the internal fluid core of the axon incredibly fast.
It's a passive fluid transmission then.
Yes, but because it's passive, the signal degrades as it travels.
Weakens.
Exactly. But just before the voltage drops below the critical threshold where it would die out completely, it hits the next bare gap, the node of Ranvier.
And that's where the sodium channels are.
Right. The dense cluster of sodium channels there senses the remaining voltage. They snap open, sodium rushes in, and the nerve regenerates a brand new full-strength action potential.
Okay, so it's essentially like a biological signal fire on a mountain peak.
Oh, I like that.
Yeah, you don't have to bring the entire forest down to send a message across the continent. You light a fire, the light travels passively and instantly through the air to the next mountain, where a lookout sees it and lights a fresh fire.
That is a highly accurate mechanical analogy, actually. The signal isn't jumping through the air, it's being rapidly transmitted and then actively regenerated at specific spaced intervals.
And what makes saltatory conduction so elegant is the energy conservation. The neuron doesn't have to expend ATP pumping sodium and potassium ions back across the membrane along the entire length of the cell.
Right, it only does the heavy metabolic lifting at the nodes. You get maximum conduction velocity with minimal metabolic cost.
Which brings us right back to the living room and the stub toe.
The dreaded toe.
If myelination is this masterpiece of speed and energy conservation, it really begs the question, why didn't evolution just myelinate everything? Like, why do we still have that agonizing 1-second delay for the throbbing pain?
Because even with the energy saved by saltatory conduction, producing and maintaining myelin is a massive metabolic drain on the organism as a whole.
It's biologically expensive.
Extremely. You have to sustain billions of specialized glial cells, oligodendrocytes in the central nervous system, and Schwann cells in the peripheral nervous system just to manufacture those sheaths. So the body performs a ruthless cost-benefit analysis.
So it basically categorizes the fibers based on importance.
Exactly.
Looking at the clinical taxonomy, the blunt, throbbing ache from your toe travels along unmyelinated C fibers. Because they lack insulation, they're like a slow dial-up modem. The conduction velocity tops out around 0.5 to 2 meters per second.
Which is agonizingly slow. But for diffuse, non-emergency pain, the body decides the speed just isn't worth the metabolic price of building myelin.
But contrast that with the initial sharp impact of the toe actually hitting the table.
Right. That signal travels along A delta fibers, which are thinly myelinated. That bumps the speed up to roughly 5 to 30 meters per second, like a standard broadband connection.
And then for absolute critical survival functions like proprioception, knowing exactly where your limbs are in space, or complex motor control, we use A alpha fibers.
The high-speed fiber optic cables.
Exactly. These are heavily myelinated and conduct at a blazing 80 to 120 meters per second. So the dual sensation of stubbing your toe is just a real-time demonstration of different fiber types firing simultaneously, but arriving at your brain at vastly different times based purely on their physical insulation.
Precisely. We only lay down the expensive high-speed infrastructure where split-second timing is an absolute evolutionary necessity.
But that reliance introduces a severe vulnerability, doesn't it? If our most critical reflexes and motor controls depend entirely on this delicate, perfectly spaced insulation, what happens when those glial cells fail?
That's when you see the catastrophic reality of demyelinating diseases. And the clinical literature draws a really sharp distinction between how this failure manifests in the central nervous system versus the peripheral nervous system.
So, starting with the central nervous system, the paradigm example of CNS demyelination is multiple sclerosis, right? Which Jean-Martin Charcot characterized back in 1868.
Yes, MS is an autoimmune disorder where the immune system attacks oligodendrocytes in the brain and spinal cord.
And when you strip that myelin away, you completely disrupt the biophysics of saltatory conduction that we were just talking about.
You do. The axon is left bare. But remember, it doesn't have a uniform distribution of sodium channels like a naturally unmyelinated C fiber would have.
Oh, right. Its channels are still clustered only at the nodes.
Exactly. So the current leaks out through the newly exposed membrane, and because there are no channels in that stretch to regenerate it, the action potential simply dissipates. The signal fails entirely.
Which perfectly explains a Toff's phenomenon in MS patients. Clinicians have long noted that MS symptoms can acutely worsen when a patient's core body temperature rises, even just from like taking a hot shower or exercising.
Yeah, and it's not that the heat is causing more immune damage in that moment.
It's pure physics, isn't it?
It is purely biophysical. Warmer temperatures accelerate the opening and closing kinetics of voltage-gated sodium channels.
Okay, so they open and close faster.
Right. Now, on a healthy myelinated axon, that slight change in channel timing doesn't matter. But on an MS-compromised axon where the myelin is thin or missing and the current is already barely making it to the next node.
That faster channel inactivation is enough to cause a complete conduction block.
Yes. The action potential just fails to regenerate.
Wow. Now, contrast MS with Guillain-Barré syndrome, or GBS. This is an autoimmune attack, too, often triggered by an infection, like Campylobacter jejuni, but it targets Schwann cells in the peripheral nervous system.
Right, outside the brain and spinal cord.
And it can cause rapid paralysis. Yet the prognosis for GBS is remarkably different from MS, generally speaking.
And it all comes down to the architecture of the glial cells themselves. In the CNS, a single oligodendrocyte is a massive multitasker. It extends dozens of processes to wrap myelin around multiple different adjacent axons at once.
So if one goes down, it takes a lot with it.
Exactly. If an oligodendrocyte dies, you lose the insulation for an entire network of nerves. But in the PNS, Schwann cells operate on a strict 1-to-1 ratio. One Schwann cell wraps one single segment of one single axon.
So when the peripheral nerves are attacked in GBS, those dedicated Schwann cells have a much simpler biological task when it comes to regeneration.
Yeah, they just have to rewrap their specific segment, which is why most GBS patients experience substantial functional recovery, whereas repairing those multitasking oligodendrocytes in the CNS is notoriously difficult.
That structural difference is so critical. But, for a century, neurologists viewed these diseases primarily as electrical failures. The rubber degraded, so the copper wire short-circuited.
Right, that was the dogma.
But that entire paradigm was upended in 2012 by a researcher named Klaus Armin-Nave.
Ah, this is such a brilliant discovery.
This is perhaps the most fundamental shift in the modern literature. Nave discovered that myelin is not just an electrical insulator. Embedded within the myelin sheath are specific transporter proteins, specifically monocarboxylate transporter 1, or MCT1.
And these proteins are actively shuttling lactate from the oligodendrocyte directly into the axon.
Which means myelin is actually a vital metabolic support system.
Yes. Axons, particularly long ones, are highly demanding structures, and they often struggle to transport enough glucose down their entire length to survive. So the oligodendrocyte is actively feeding the axon with lactate, which the axon's mitochondria then convert into ATP.
Okay, let me push back on the old model here, because this is where it gets really wild.
Yeah.
If you strip the rubber insulation off a copper wire in a house, the wire still works perfectly fine as long as nothing touches it.
Sure.
But based on Nave's discovery, myelin isn't rubber tape. It's an IV drip. So if you remove it, The nerve doesn't just short circuit,
Starves.
it starves.
That's the critical revelation. In demyelinating diseases like MS, the ultimate cause of permanent disability isn't just the electrical conduction block we talked about earlier, it's axonal degeneration.
The nerve fiber physically dies.
Because its food supply has been severed. Treating MS isn't just a matter of restoring electrical insulation, it requires restoring a highly active metabolic partnership.
And if oligodendrocytes are active metabolic partners, it completely shatters the old dogma that myelin is a static, inert substance.
Right. For most of the 20th century, the assumption was that myelination happened in childhood. Your brain matured, the insulation was laid down, the concrete dried, and by adulthood, your white matter architecture was essentially fixed forever.
The done-in-childhood myth. It really took advanced neuroimaging to prove otherwise, didn't it?
It did. The turning point was a 2009 study by Jan Schultz using diffusion tensor imaging, or DTI.
DTI, right. That's a specialized MRI technique that tracks the fractional anisotropy of water molecules, correct?
Essentially, yes. Water diffuses more easily along a myelinated axon than across it because the fat in the myelin blocks the water. So by tracking that diffusion, researchers can actually map microscopic changes in white matter structure.
And Schultz did this with adult humans. He mapped their baseline white matter and then had them practice a complex, novel motor skill they had to learn how to juggle.
Right, a totally new skill for adults.
And after just a few weeks of deliberate practice, the DTI scans showed measurable physical increases in the fractional anisotropy in regions of the brain associated with grasping and visual motor coordination.
The white matter physically changed in adults.
But DTI is correlational, right? To prove a direct causal link between motor learning and new myelin, science needed to actually manipulate the biology.
And Ian McKenzie and his colleagues provided that in 2014 with a really brilliant study on mice. They put adult mice on a complex running wheel, a wheel with irregularly spaced rungs that required the mice to learn a very specific, challenging gait.
But the researchers didn't just observe the mice, they genetically manipulated a subset of them. They selectively blocked the formation of new oligodendrocytes in the adult brains.
Yes, they physically prevented those mice from generating new myelin.
And the result?
The mice with blocked oligodendrocytes were completely unable to master the complex wheel.
Wait, really? Not at all.
Not at all. They could run perfectly fine on a standard wheel, proving their baseline motor function was totally intact, but they could not learn the new skill.
Wow.
This definitively proved that adult motor learning doesn't just rely on synaptic plasticity, changing the connections between neurons. It actively demands myelin plasticity. The brain must manufacture new insulation on demand to lock in complex new behaviors.
So it's like a home renovation. It's not like the house's electrical wiring is permanently finished the day we become adults. The brain is literally adding new insulation to specific wires based on which rooms or skills we use the most.
Exactly.
But how does the brain actually know which specific axons to myelinate during learning? Like if you're practicing the piano or trying to fix your golf swing, how does a precursor cell in your brain know exactly which motor pathways need the structural upgrade?
It comes down to cellular communication. When an axon fires repeatedly, like when you practice that piano scale over and over, it releases neurotransmitters along its length, specifically glutamate.
Okay, glutamate.
And the oligodendrocyte precursor cells, or OPCs, scattered throughout the brain have glutamate receptors. They literally sense the electrical activity of the axon.
So the active axon is essentially broadcasting a chemical signal saying, hey, I'm being used constantly, I need an upgrade.
That's exactly what's happening. The OPCs migrate to that active axon, differentiate into mature oligodendrocytes, and wrap it in new myelin.
It's activity-dependent wiring. The nervous system acts as a master electrician that never clocks out.
And it's not just blindly adding thicker myelin to make everything as fast as possible either. That's a common oversimplification. Recent research shows the brain uses myelin as a highly tunable parameter. It actively adjusts the thickness of the sheath and the length of the nose of Ranvier to fine-tune conduction velocity.
Wait, adjust? Why would you ever want to slow a signal down?
For synchrony.
Synchrony?
Yeah. Think about it. If you are catching a falling glass, your motor cortex has to coordinate inputs from your visual cortex, your proprioceptive feedback, and multiple different muscle groups.
Right, it's a huge coordinated effort.
And those signals are originating from different physical distances in the brain. If they all traveled at the exact same maximal speed, they would arrive at the target out of sync.
Oh, wow.
So the brain specifically tunes the myelin on shorter axons to slow signals down and sickens it on longer axons to speed them up, ensuring all impulses arrive at the muscle at the exact same millisecond.
It is about perfect temporal precision, not just raw speed. It really puts everything into perspective, doesn't it?
It does.
I mean, from the sheer size of the giant squid axon fighting internal resistance
Yeah.
to the evolutionary brilliance of mammalian saltatory conduction, the nodes of Ranvier actively regenerating signals, the metabolic tightrope act of fueling long nerves with lactate. We've seen how devastating it is when that system starves in MS and how remarkably adaptable it is when we force it to grow by learning something new.
The human nervous system is an infinitely dynamic living tapestry. And as you are listening to this deep dive right now, processing these complex physiological concepts, the active axons in your own cortex are releasing glutamate.
That's amazing to think about.
Your precursor cells are migrating. Your oligodendrocytes are quite literally feeding your nerves and potentially remodeling your white matter to encode this new information. Your biological infrastructure is upgrading itself at this exact moment.
Which leaves us with a truly fascinating implication to ponder. If myelin plasticity is a lifelong process, and if our nervous system uses it to specifically tune the microscopic timing of our neural circuits, it forces you to rethink human potential.
How so?
Well, could the difference between a highly skilled athlete and a once-in-a-generation Olympic gold medalist not just be in their muscle mass or even the speed of their synapses? Could the absolute upper limit of human performance ultimately come down to the microscopic, perfectly tuned timing of their myelin insulation, meticulously built up through thousands of hours of deliberate practice?
It strongly suggests that greatness isn't just something you perform. It is quite literally something you insulate into your physical architecture.
It makes you realize that the static imagery we usually rely on, the plumbing, the wires, the passive tubes, completely fails to capture the reality inside us. Your brain isn't a fixed electrical grid. It's a constantly adapting, metabolically hungry masterpiece of engineering. And that is a biological truth worth holding on to. Thanks for joining us, and we'll catch you on the next Deep Dive.